Okay, welcome everybody. We got all committee members are here, which is all we need. Nice to see the rest of you here. Happy to introduce Adam Chouinard for his public thesis defense. Adam did his undergrad at University of New Hampshire and stayed there on to a master's, working on Plethodon and salamanders for a master's degree. Arrived here at OSU in 2010 and did some field work associated with his degree, has done a lot of computational biology and quite a bit of biochemistry. So he's going to tell you all about that today. So Adam, welcome.
I pretended to be a biochemist.
And I'm going to turn off the top.
So yeah, let me also echo that and say thank you all so much for coming. I really appreciate everybody showing up on Friday of finals week here. And as you've gathered, I want to talk about pheromone communication in a really interesting group of salamanders. And we'll kind of ask this general question. It's really just how are they communicating in the broadest sense via these chemical signals? So I'll sort of provide some context for you as we march along our talk here and discuss communication in general, chemical signaling in general. And then we'll talk about why I'm so interested in variation and how that relates to communication. Provide some background on the species in question. And then we'll look at how these signals are composed, what actually makes these signals, and how they vary at multiple levels of scale. We're going to get into all this, so I won't really say too much about it now. But then we'll sort of figure out what we can take away from this and where we should go next. So our story begins, or I should say my story, it's your story now, on Earth here, of course, in the eastern half of North America, in the deciduous forest there, and specifically this deciduous forest, because this right here is actually the house to which I was brought home from the hospital. So we're really starting at the beginning here. And some of my earliest memories, well, some of my earliest memories are wandering around and checking out the forest outside the house there, and turning over rocks and logs and just kind of rummaging around in the leaf litter and finding these individuals, the red-backed salamander, Plethodon cinereus, a really interesting relative of ours. And this is actually the most abundant vertebrate in terms of biomass in the eastern United States, or the eastern half of North America, its range. And many years later I'd go on to study it, and then a few more after that I'd go on to study this species right here, Plethodon shermani, the red-legged salamander, another relative of it, so a closer relative. And both of these have served as interesting systems into which we can ask this question, is how do amphibians communicate via chemicals? But they've served as models for that in kind of a different way. Plethodon cinereus has been a model for how chemical signals are involved in sort of social behaviors and what roles they play in their social and reproductive behaviors. And then Plethodon shermani, we actually have looked at the biochemistry of a particular gland that we're going to talk about here. And so we know more about the biochemistry in this case. And I want to look at both and kind of take a comparative approach here, sort of across multiple levels. And so my story didn't really just even begin there. It began here, right, as it does for all of us. And all of our collective journey really goes back a lot farther than that, right? This is a tree of life, or phylogeny. We'll see a couple of these today. It's just like a really big, big family tree of life on Earth, right? When it plays out over millions of years. And in case you're wondering, that little tip right there is us in a very large tree of life, right? So whether we're looking at how cells work and how cells communicate with each other, or how whole ecosystems function, right? One theme that really jumps out to me that's really fascinating is communication. Because different entities need to communicate with each other in order to sort of just carry out the play of life, right? And so we'll kind of paraphrase this. This is a definition I like. Essentially, we can think of communication as an action on the part of an organism. It's going to affect the probability of other entities' actions, right? And I should say even organism, really any entity, because you can think about this across levels. And so if we have a sender of a signal here, say an odor that makes its way over to a receiver here, who picks that up, and if this affects even the probability of their behaviors, then we can say the communication has taken place under this definition, right? And since this was a scent and odor, we would say that this is a chemical signal. And specifically sort of within that category, because these are the same species of the red-legged salamander here, we would say that that's a pheromone. So we're going to look at pheromones and actually chemical signals in general in this really interesting group of organisms, the family Plethodontidae, the lungless salamanders. They compose actually like 2/3 of all the salamander species. So they're a very large group. They are the lungless salamanders, as I said, kind of ironic because they're also some of the most terrestrial salamanders that there are. And I'd love to talk about a lot of their biology today because they're really kind of cool in a lot of ways, but we're going to focus on the role of chemical signals. And this sensory modality is very dominant for these salamanders. And chemical signals facilitate a lot of functions between species, so inter-specific effects, you'll hear that a few times today, between different species, but also within the same species, so the real pheromones over here. I've been interested in my research over the years in things like territorial scent marks that they place to defend their territory. But pheromones are also very important, and a lot of the research has taken place in this system on their role in facilitating reproduction, via like locating mates, actually assessing their quality, and facilitating the courtship ritual itself. So they're used directly in courtship. And we'll look at these things today. But in totality, these signals convey a lot of complex information, like the species of the sender, the sex of the sender, how big they are even, which individual it was. If you're a female, whether you're carrying eggs and you're reproductive. Things that are condition dependent, right? Like the quality of their diet, their parasite load, if they're parasitized. Whether they've suffered an injury, like a loss of a tail. So pretty complex information transfer, actually, in a sensory modality that's very different from our own. And so we'll employ this working hypothesis to convey complex information, right, and to carry out different behavioral functions. Chemical signals, or really any signals, need to vary in their composition, right, in order to do that, to have different effects. And my research has explored the causes and the consequences of this variation in the way that these individuals communicate via chemical signals in plethodontic salamanders. And we're going to look at that today at multiple levels of scale, actually. We'll sort of zoom way down in on the body of a single individual, look at how different regions communicate differently. We'll look at members of the same species, so sort of population level analyses. And then we'll zoom out and look at different populations of the same species and between different species, and even whole lineages of this entire plethodontid, this group, their family tree. So why does variation matter, though? Why am I going to be talking about this right now? It actually is very important for evolution, right? Variation is sort of the fuel for evolution. And so there are proximate and ultimate consequences of variation, sort of the evolutionary jargon. But think of that as like the mechanistic consequences and then the evolutionary consequences. So in the former, variation in the signal is what literally dictates its meaning, right? The way the signal is composed dictates what it means. But if these signals are involved in reproduction, these can also have evolutionary consequences, right? If they sort of vary in some sensory space, and this plays out over time and different sublineages start diverging in that sensory space, you can have speciation events take place. So let's look at a real example of this, and actually a really good one comes from amphibians. A really cool genus here, Cynops newts from Asia. And so this is actually the first peptide or protein pheromone from vertebrates that was identified. Its name is cedefrin. And it's a very small peptide, 10 amino acids. And it's produced by males only, and it is a very potent mate attractant. Females hone in on this when they're reproductively active. And because of the fact that females don't produce it and males do, we can say that it encodes the specificity, if you will, of the sex of the sender. They can tell that this is a male signal. And because of the fact that different species of the same genus also differ in the signal from essentially the same gene, ultimately, in this case, it's actually cleaved from a larger precursor, but the difference between two species of the same genus is just 2 amino acids, and yet they carry out species-specific effects on receivers. So in this case, they've diverged in this one little snippet of their sensory realm, right? So what about plethodontids, though? How do they communicate, and how might this play into the role here, into this system? Well, they have different regions of the body that communicate, or the hypothesis here is that they're adapted for different functions based on behavioral experiments. A big one is the mental gland. This is where almost all of the biochemistry in the system has been done, and we'll talk about it today. It's used in courtship, and we'll see its role in just a second here. But they also have different regions of the body that have specialized glands, anatomies, and functions, like the bottom of the tail here, posterior to the cloaca. And they actually place these active scent marks down in their territories to advertise them. There's glands within the cloaca, actually several of them, that are responsible for spermatophore production, but they're also responsible for, where they appear to be anyways, things like more general social and even territorial signals. And then there's this dorsal tail base, which is a little bit of a conundrum. We don't know a ton about it right now, but it appears to be also potentially involved in courtship. And again, it has different gland anatomy, so something's going on there. And it might be making courtship signals. Let's focus on the mental gland, because again, that's where most of the work in this system has been done in terms of the biochemistry. So this gland is used in courtship, and the way it's used differs between different lineages, and we'll look at that. But essentially, courtship consists of what they call the tail straddling walk, in which the female follows the male here in the front, and he sort of she straddles his tail, he undulates it, and periodically, as he's kind of marching forward, turns around and applies the secretions of the mental gland to the female. And the effect is to reduce the duration of the courtship ritual. So this has been used kind of as a proxy for female receptivity. But the way they deliver the pheromone, again, differs between different species. And so this group, the ancestral state, so it existed earlier in time. Males have these, what they call protruding premaxillary teeth, and they're on the front of the snout, and females don't have them. And they use these to actually scratch at the female's skin and rub the mental glands over the wound. So the hypothesis here is that they're actually vaccinating the female's bloodstream with these pheromones. Kind of neat, right? But a derived state within this lineage, the plethodontity, is they actually develop these huge, massive, tumor-like mental glands, right? And they make even larger amounts of these proteins, and they turn around, and it's much the same scenario here, but they apply these secretions from the mental gland directly to the female's snout. And it acts through the olfactory pathway, and we know that actually experimentally. It's stimulating neurons in the olfactory pathway. But what's actually in it? Well, before we can answer that, we should all kind of get on the same page, right? So we all know where we're going, and we can clear up some of the language. You've heard a gene for this, and a gene for that, a gene for this. But what genes really do, mostly, is encode for the sequences of proteins based on the sequence of nucleotides that make up your DNA. But you don't get there directly. There's an intermediate molecule, a single-stranded nucleotide molecule called messenger RNA, mRNA. And I'll refer to these as transcripts because they're produced via the process of transcription. Then that signal, that message, or coding sequence, I might use that word here, talking about transcripts, get translated into the protein that actually exerts its functionality, right? So this might look something like this. This is kind of one of our best simulations of this, using real structural models in real time, actually. These are all free nucleotides swirling around, and this is a big protein complex that specializes in this task. Looks like it's probably like the world's most enjoyable roller coaster ride, according to this video, right? Look at that. But it's basically reading the sequence of nucleotides in the DNA and turning them into an mRNA molecule that matches that coding sequence in the DNA. That finished mRNA molecule gets processed a little bit, but it gets taken out of the cell, out of the nucleus of the cell, which is where the DNA is housed. And it finds a ribosome. And a ribosome is a big complex of different proteins and also RNA molecules of a different type. And it reads the sequence that came from that gene sequence ultimately after it gets processed down a little bit and matches up the corresponding amino acids that make up a protein, right? Now, this is kind of a different chemical, but it's really coming from that same ultimate message that was from the gene, right? The protein's going to fold up in three-dimensional space and carry out its effects on whatever functions it plays, whatever functions it employs, right? Or it's used for. So now, we'll talk about transcripts, we'll talk about proteins, but what's in this signal here? Let's go back to our question. Over the entire plethodontid phylogeny, there's really three main proteins that make up the mental gland secretions. Cephalin precursor-like factor, or SPF, plethodontid modulating factor, or PMF, and plethodontid receptivity factor, or PRF. This was the first one identified from this system. You'll notice this gene, this is the precursor that cephalin molecule is cleaved from in the different family of newts. So it's a very ancient salamander pheromone, going back hundreds of millions of years, this gene. And like the other genes that have kind of come after it, it has duplicated many times within and kind of between these different lineages. So this is a common theme here. There's many, within an individual male and within an individual population, there can be many sequences, many variant transcripts that get translated into different protein isoforms. So when I say that, I mean it's basically the same protein. It really has the same overall sequence. But there are regions that vary, And so these are called isoforms. They end up being slight sequence and then therefore structural variants of one another. And so if we kind of put all this together, we can build some characteristics of the mental gland pheromones, and they're all present in multiple isoforms in a given male. These are the result of multiple gene duplications, as we can infer via phylogenetics. And they've diverged in sequence between species, and actually even between these numerous gene duplications, right? And we can look at sort of evolutionary signatures of selection, and they show signs of very strong positive selection. So this really just means that they're changing much more rapidly than we would expect by chance, right? There seems to be some selective force here, and the hypothesis is that it's presumably the result of sexual selection. So I won't really go into the details of these values very much, but for those of you who kind of are familiar with these things, there they are for you. But what this really is showing you is that this sperm lysin has been kind of a paradigm of a very rapidly evolving protein, and higher numbers indicate selection, positive selection. And PRF has higher numbers than that. These are among the most rapidly evolving proteins that we know to science to date, right? They're up there. So why? What's going on there? Well, we're going to look at how these signals vary at multiple levels, and we're going to get to each of these in turn, so let's just focus on the first question here. Let's look within one species. How do different individuals of that same species vary in the composition of their mental gland secretions? Well, we need to get some samples, and so we take a salamander, put him in some anesthesia. So this is ether. He goes on a little spirit journey and goes under the microscope, sees a bright light at the end of a long tunnel, and you can take some tissue. Now they survive from this just fine. We're just going to snip off these little skin glands right here. We're going to look at the mental gland specifically. And I should say this is P. cinereus in these pictures that I have, but we're actually going to do this in P. shermani because we have a very well characterized mental gland protein scenario here. So you can take those glands, put them into a solution that actually allows them to secrete or causes them to secrete their products. You can scoop all these up from that solution and purify them a little bit and analyze them. So this is the method that we're actually using, but really don't worry about the details too much. We're just going to separate these molecules in time and in space from one another. We're trying to get individual components of this complex mixture. And then we're going to look at how much of it, of each of those components, is present. And when we do that for the mental glands of P. shermani, this had been done. This had been done by collaborators of mine at the University of Louisville. They figured out what all these different things are and refined this technique. And you can see that this is PMF, and they're making tons of isoforms. Males a single male make dozens of isoforms of PMF. This is kind of a neat tangent, but it's a protease inhibitor that's presumably protecting all these from degradation. And then this is PRF, and the idea was that they're making 3 isoforms. B, C1, and C2 is sort of the terminology we're going to use here, but 3 isoforms. But we'd only ever looked at whole pooled samples, the entire population signal at once. And it turns out that this is obviously an oversimplification, because I want to know how much do these individuals in this population vary, because that's what matters for selection, right? That's what selection is actually going to be acting on, night to night, when these individuals are courted. So when you do that, you look at, this is like 104 individual mental gland secretions. This is PMF varying all over the place. We'll just focus on PRF varying all over the place, right? PMF had been described in the literature just based on its sequence as a hypervariable gene group, right? And you can see that's true for their levels of expression as well. And PRF, there's a lot more going on in the range of PRF than we had anticipated, because individual males can be making many more than 3 isoforms. So there's more going on here, and there's substantial variation in the presence of these isoforms, as you can see in a second here. We can also kind of quantify the amount of the individual components a little bit more statistically in this case, because we can account for the variation between individuals. But PRF and PMF are making up over 80% of the actual secretions, and more PMF than PRF. But we see that there's, between individuals, substantial variation in the number of PMF peaks, and this is the number of individuals, or the percent of individuals that have each peak. And they range from like down to 15 up to 40, right? In PRF case, they can range from those 3, mainly the main isoforms that we'd seen, but they can have more than that. And there's sort of more commonly that they have 4. And when we look at individual male profiles, this is the raw chromatogram again, is what we call these. You can see that this is kind of a typical one that we'd always thought was present. It's the most common. But there's lots of variation. We're missing B, we're missing C1, we're missing C2. We've got extras down here that have never really been accounted for. And this could be perceived by females very differently than just this, right? So if we focus in on PRF, and let's just restrict ourselves to the 3 main isoforms for now, we can also look at how their levels of expression vary, not just their presence and absence. And that's what's plotted here. So it's sort of a little bit counterintuitive first, but this is basically just each of the 3 isoforms and their proportion from 0 to 1 in the overall mixture. So this is the entire variable space. This is all of the possible permutations of these 3 molecules and their expression. And you can see that males are taking up a huge amount of this possible space. These are individual male profiles, right? But when you look at the whole population, there's a more common profile. And this is what we've typically seen when we look at whole population levels, actually. And what's really fascinating is there's one other study from a while ago that looked at whole population samples and looked at basically their chromatograms, their profiles. And there's evidence that different populations of this species complex occupy different regions of this graph, which is exactly what I would have predicted, actually, if they're using these as sort of reproductive isolating barriers, right? So they may be diverging into different zones of this sensory space, right? Kind of like what we talked about. It's really neat, but of course we need to sort of revisit that and experiment with it more. So what can we take away from this? Well, there's substantial intraspecific variation in mental gland pheromone composition, made up by the isoform number of these individual components, and also their levels of expression, right? And there's evidence that there could be population-specific isoform ratios that might indicate divergence in these courtship pheromones. And overall, these levels of expression could be a whole other level of signal complexity beyond just the sequences themselves, right? And there's actually numerous minor components in the range of PRF. And just because they're lowly expressed, it doesn't mean they're unimportant. Or to undo a double negative here, lowly expressed things can be important for these. We know that from other systems, where minor components, if you will, are actually essential for encoding things like species specificity in the signal. Okay, so let's zoom out a little bit. We'll look at different populations, and then we'll look at different species and different lineages even. In one of these key pheromones, plethodontic receptivity factor, PRF, that one we were just looking at. And we need to know a little bit about their relatedness, right? So there's a lot going on here, but don't worry too much about a lot of this right now. Let's just walk through it. Ignore all the species names. Back in the day, when plethodontids were diverging away from the other families, everybody had SPF. Remember, that one goes way back. And PMF. Everybody had a mental gland and protruding premaxillary teeth, and they used that dermal delivery mode of pheromone delivery. You get speciation events that lead to the western plethodon diverging away from the eastern plethodon. And this group in particular is a very rapidly radiating clade when you look at the actual rates of lineage accumulation, as others have. And so this is when we have PRF appearing. In the eastern plethodontics, not present in, say, the western or the western redback salamander. And within the eastern plethodon, you have these basically 2 main lineages with kind of a weird transitional state where they do different kind of combinations of these traits. We have the ancestral group, which is the dermal delivery, which includes Plethodon cinereus, by the way. And then we have a highly derived olfactory group over here, which includes Plethodon shermani, as you can see here. So that's sort of our roadmap for how these species have evolved. But how has PRF evolved over the course of this rapid radiation? What's going on between these lineages, right? So we can take all the known sequences. There were like 200 of them on file in our databases from previous researchers who've investigated sort of the evolution of these proteins. I actually went and collected a totally different population of P. cinereus. Their one on file kind of was from Virginia, down in the south. And I went back to where I had studied P. cinereus before and got more samples from New Hampshire, a pretty removed population, right? So we could argue about species designations, and people actually have in this exact species. But we can line up all the sequences according to sort of their homologous position, the position in the protein that corresponds to the sort of previous ancestral position in the ancestral proteins. And look at how conserved they are, and look at other things like how rapidly certain sites are changing. And we can map out basically their conservation. So high degree of conservation means everybody's got the same amino acid at that site. And low conservation would mean that it's variable, right? That site varies a lot. And we can actually visualize this in 3 dimensions. I've made a model here of what PRF probably looks like based on its conserved fold with other known relatives whose structure we do know. And then we can map out very variable regions or very conserved regions and see what it actually looks like in 3 dimensions. You can see across all the PRF sequences, there's regions that have pretty high, or rather low degrees of conservation, right? So high variation in these 3-dimensional structures. And they occur kind of out on the loops and out on the terminal regions of this protein. But we can also look sort of between different groups, right, and look at comparisons there. We can look at the dermal delivery group and what a PRF there looks like and how much it varies within that species compared to an olfactory group and what the PRF looks like there and how it varies, right? And there are big differences, right? We have the potential for quite an add-on to this protein structure in this lineage that isn't present in the olfactory delivery group. And there's also more intraspecific variation with these names. And if we just look at that, we can zoom down to the population level again and look at different populations. So those Virginia population that was kind of in the databases versus the new sequences that I got from New Hampshire, which, by the way, include some really weird PRFs, actually, that weren't present in their kind of closer relative, right? But this is just the tip of the iceberg, actually, because there's a lot more level of sort of structural variation here. Because PRF wouldn't just look like this in the dermal delivery plate. Same thing, just all the colors stripped away. It actually looks probably something more like this, because these are large carbohydrate groups that get added on to the structure after it's been translated. And we call this process protein glycosylation. And there's 2 different ways that apply to the case of PRF anyways, in which glycans, as we'll call them here, carbohydrate groups, sugars, can be added on to these. And they occur at known motifs. Those motifs differ a little bit by the different types of glycosylation. But we can look at that. And the observation that started this whole thing off is we'd run out, my collaborators at the University of Louisville and I collected my samples here, compared them to the P. cinereus from Virginia, and compared them to P. shermani. So there's a lot going on here, but we'll walk through it, right? This is same thing, but we just stain for proteins. And this is when we stain for carbohydrates, and then we overlay the 2. So if we focus up here now, we're just separating out proteins based on a different property in this case, just based on its size as they migrate through this physical matrix that I'll call a gel. And you can ignore the outgroup here, they don't have PRF. But if we just look at P. shermani, PRF should appear right here, and it does, at 22 kilodaltons, where it belongs, its size. And it's not glycosylated, as we can kind of see from the gel, but also based on other proteomic observations that my collaborators have done. And when we look at P. cinereus, though, it seemed like, whoa, there's these weird other components that they're not making. Maybe they have a whole other pheromone. It's not a whole other pheromone, it's PRF, but they have these huge sugar groups attached to them, and they don't make it as far through the gel. And what's really interesting to me is that there's evidence for it anyways, substantial intraspecific population variation here, or intraspecific variation here, in terms of which kind of variants of these glycosylated PRFs they're making. So I want to know, all right, does this observation, which are from individual species, do they really apply to their whole lineage, all their other closer relatives, right? Are these representative observations? And so we can use some of the tools at our disposal, because it's hard to get protein samples, right? Let's use other tools. And we can look at the protein sequences themselves and try to predict this and see where these motifs occur. So again, kind of a lot going on. This is just a signal peptide here. You can kind of ignore that. We've lined up all the protein sequences. This indicates there's amino acid there, the dark. And then there's gaps in some of these sequences as they evolve. There's deletions and insertions. And we have the dermal delivery group up top, then kind of the transitional state, and that olfactory delivery group. Now in both cases here, I'm going to map on the possibility that there's glycosylation. Whether it's O-linked in kind of the blue shades, or whether it's N-linked in the reddish shades, right? And in both of those cases, blue or red, O or N-linked, whether it's the light shade means that, well, there's a possibility that it could be glycosylated. The motif is there. And whether it's darker, say dark red versus orange, means that it's predicted to be glycosylated, actually. The structure seems appropriate for it. So I'm predicting, if those observations are representative, I should see lots of potential for glycosylation in the dermal delivery plate, but not the olfactory delivery plate. And that's exactly what we end up seeing, right? There's a let's look at the N-linked motifs. Up in this dermal delivery plate, these two we know proteomically from other experiments that my collaborators have done are glycosylated. But there's also other regions. There's actually 4 sites overall that can be glycosylated with N-linked glycosylation, but these motifs have been completely lost over the transition to olfactory delivery. They don't have that potential, explaining our observation in the gel, right? Similar situation with O-linked glycosylation, where we actually have prediction, and again, one of these sites at least was shown to be O-linked glycosylated in P. cinereus by my collaborators. There's stronger predictions there, right? There's almost no potential for there's really no observation of glycosylation in the olfactory group, right? And with specific reference to the N-linked groups, they've been completely lost over this transition, right? And there's substantial variation within species, right? Hence the multiple patterns that we see in the gel for a given species, a given sample. This is the New Hampshire population with an entirely different variant. And even at the 2 main sites here, they differ in their predicted patterns. So maybe something's going on in this New Hampshire population where that site's no longer as suited for glycosylation. So this is just the first look, right? These are just some predictions, but they're hypothesis generators, and they've actually helped us corroborate some of our observations from the gel. And we can also look at how these sites have evolved in a more phylogenetic sense, right? So again, don't worry about all the tips out here. We'll walk through them. This is like the same thing, but it's just the gene tree for PRF, not species trees. And we have a whole other lineage of PRF isoforms called the PRF-B isoforms in the dermal delivery group. And they all branched away from the PRF-A isoforms that everybody else has, including the dermal delivery group. So as a whole, they have A and B isoforms, this big part of this phylogeny, right? And then here's kind of our intermediate group we're sort of glossing over for the sake of time today. Collapse down the entire olfactory plate because they're not glycosylated, right? So we're mapping that trait on here. So let's look at this. That's our same phylogeny for site 1 and site 2. And the colors are the same thing, but kind of ignore the difference in shade now. We're just going to look at the presence of these motifs. And we can infer quite a few things from these types of analyses. Well, they were present in the ancestors of all the PRF-A isoforms, but not the B isoforms. So that's kind of something already. And they were lost at different stages over this transition to olfactory delivery. So they were lost independently, but pretty much in rapid succession here, right? And we can do the same thing for the 3rd and the 4th site, which show up a little bit less often. So I'll kind of refer to them as alternate. But we see some neat things here too, right? They evolved independently of one another and in different isoforms. This site 4 occurs in PRF-B isoforms. It's the only time that happens in PRF-B isoforms. And that includes the New Hampshire P. cinereus sample as a representative of that as well, right? Okay, a lot going on there, but what might it actually mean in reality? And this is where we get into sort of hypothesizing, right? We're continuing to hypothesize and looking at some evidence for this hypothesis to see whether we should continue to investigate it. And we know that PRF has a relative that's pretty well understood, and that protein's name is interleukin-6, or IL-6. And PRF is an IL-6 type cytokine. It's a part of this bigger level of a gene tree. It's a relative of this protein. And we know its receptor. And this is a solved 3-dimensional structure. This is IL-6, and these gray shades are different parts of this receptor. Now these are mirror images of one another, so I'll kind of just gesture to one. We have one region of binding right here with this IL-6 ligand. I might use that word here. That just means it's the protein that's actually stimulating this receptor. We have a site 1 binding. We have a second binding site right here. And again, these are experimentally known in IL-6. And we have another 3rd region right here, actually. Now I've mapped on the known O-linked glycosylation sites and the known N-linked glycosylation sites. For IL-6, it is glycosylated, but they don't affect binding, and we know that experimentally. I'm going to just tip the molecule up on its side. Now I'm looking parallel to the cell membrane, so like this would be the cell membrane. Those O-linked sites are way up and out of the way of all those known binding sites. And the N-linked site looks like it could be in the way there, right? But if we flip it over like that, now we'd be like lying on the cell membrane looking up. It fits right into these little binding, non-binding pockets, right? They're just out of the way. And again, we know experimentally they don't affect binding in IL-6. So now let's tip it all the way back up. We'd be looking down at the cell membrane. And let's overlay PRF, a closer relative of this IL-6 ligand, right? We can see what it might look like in the ancestral receptor pathway, okay? Now let's map its glycosylation sites. O-link sites and N-link sites in red. And they occur right smack dab in the middle of these binding sites. Unlike IL-6, I'll tip it up at about a 45-degree angle. These are crucial binding regions of this protein, of receptor complex, right? And let's keep flipping it. You can kind of see it. It's hard to sort of see this, but when you zoom way in, I mean, they are right smack dab in the middle. So let's flip it all the way over again. You can kind of see that from the bottom view now too, right? They're right there where those binding sites occur. So it's a good hypothesis that these might have targeted these binding sites for some reason, right? I have my suspicions. And if you think about what the structural consequences of either one predicted glycosylation site or two, so maybe different patterns of glycosylation, these could have profound consequences for receptor binding when you think about trying to fit that new weird shape that wasn't there in IL-6 at that spot into that receptor binding region, right? So there could be profound structural consequences, right? But we need to examine this. It's a neat new thing to look at. So what can we take away from this? Well, we looked at intraspecific variation, like between P cinereus, and there's variation in the sequence and the structure, as we kind of knew already coming into this. And when you think about the role for glycosylation, it's possible that males could be making many combinations of variants. I knew that was going to happen. I've been using this thing for like days. And I don't have...
I have a pointer. So
pop those out really fast. So if you think about PRF glycosylation now, it's possible that they could be making different combinations of isoforms, right? And different combinations of glycosylation patterns, right? So there's a lot of, and we saw that in the gel. So there's a lot of intraspecific observations, variation, right? Come on, buddy. Did I put them in wrong? I totally put them in wrong. Do that again. I have short fingernails. So, and we saw that in the gel, right? There's differences in patterns. There we go. I like the little clicker though. So. Okay, we're gonna just kind of go on the fly here. Come on, buddy. All right. Bob, I think I killed this thing of yours once and for all. Okay, I'll use that actually. So, let's just see if this will work. Nope. Okay. What else did we learn? Sorry. I knew something was kind of going too smooth here. There's also substantial interspecific variation in sequence and structure, right? Like the difference between P. shermani and P. cinereus. And we also know, right, PRF was glycosylated in the ancestral state, that dermal delivery clade, but lost in the olfactory group. And these are associated with very different target tissues, right? The bloodstream versus the olfactory system, very different targets. And it's a reasonable hypothesis when you see that level of coincidence, right? That natural selection or sexual selection is at play here. And it could be that it's adaptive in the ancestral state. We also know glycosylation evolved multiple times in ancestral lineages, and they target key binding sites in the ancestral receptor pathway, which is kind of neat, a whole new thing that we could look at, right? And really overall, there's a whole other level of signal variation here that we didn't appreciate before because we hadn't had that observation. And these were already really variable signals, right, when we look between species and within species. There's a whole other level, one that, just based on the nature of glycosylation, could be very, very sort of plastic, if you will, very variable. So let's sort of also now zoom way in on the body of a single individual. We'll come from one extreme and zoom way down in and look at how individuals vary within themselves, right? How do different glands vary in pheromone production to carry out different behavioral effects or convey different types of information, right? And so we talked a lot about the mental gland, but what about some of these other cool glands that we've been studying for decades at the behavioral level, behavioral experiments, and they do really neat things for these salamanders. They're very important for them, but we don't know anything about what they're actually making. So let's take a look for the first time here. And we'll revisit our little schematic here. If I'm interested, all right, what types of proteins maybe are these tissues making? I could do a proteomic approach and try to scoop up the proteins themselves, and that's definitely something you want to do because the proteins are what matter for function, right? But a way that's a little bit easier to take a more comprehensive view is to do what we call RNA-seq. And we can basically just scoop up all the transcripts, all the mRNA itself, and sequence them all sort of at the same time. These are some very powerful tools we have. So the way that we'll do this is, let's say we have just a single strand here that could encode some protein of interest, and let's say this is a part of a transcript. And because they're present in multiple copies, and we fragment these molecules up sort of randomly and sequence known lengths of them, you get what we call reads, little snippets of this sequence, right? And because there were many copies and they were sort of starting in different places, they line up with one another, right? Based on this overlap between them. And based on that overlap, we can reconstruct from our reads the sequence itself, right, of our target. Now this would be like, in the human genome, we have like over 3 billion nucleotides that make up our genome. Kind of neat factoid for you. That'd be like a book that's 3 billion letters long. Let's take out all the spaces. And we have thousands of copies of that, right? And we, just starting in random places, shred them up into little 100-letter long reads and dump them into a swimming pool, and then try to take those reads and those millions of reads and reconstruct them based on their overlap, right? It's a huge computational challenge. But we have high-powered computers. We're pretty sophisticated primates. We're really proud of ourselves. And so we can do this relatively well. And so this is called de novo transcriptome assembly in this case, right? In my case. Because I'm not using a reference. We're just using the data from the sequences themselves. And I'm trying to target all of the expressed mRNAs, the entire transcriptome. And so I took gland tissues, and we need to get the transcripts themselves, right? So we surgically excise the glands, we get the mRNA out of the tissue, get the rest of the tissue away, and we convert this into actually DNA because it's more stable. That's what we sequence. For those of you who do this kind of thing, this was done on a HiSeq 2000 with paired 100 base pair reads. We got to do some quality control with our data now. And then we assemble it. We did that in Trinity. And then we need to annotate it, and I'll look at what those actually mean. So assemble is we actually build the transcripts back from the reads. And you get a lot of metrics. Yes, this is kind of a sadistic joke because who wants to just dive through like dozens of numbers, right? But the point here is you get lots of different sort of metrics. You look at these things from different angles to see, is this the best transcriptome that I can make? So you do it, in my case, dozens and dozens and dozens of times. And I sort of ended up with one that I was kind of happy with. And there were some kind of quality issues with the data, but you work with what you got, right? And it's okay. We have a lot that we can tell from this already, right? At some point, my OCD was kicking in. But we ended up with like over 90,000 transcripts. Trinity was trying to actually cluster these down into what it thinks are genes because individual genes can actually encode multiple transcripts. It does an okay job, but they're not really that accurate. And then we can take our reads, which represent, in terms of their number of the different transcripts, they represent basically how highly expressed was that individual transcript in my different tissues. And you can then map them back and kind of count up how many transcripts were present in your tissues. And you can use this for a lot of different things, like looking at how the glands actually differ in their expression. But you can also use it to sort of hone in a little bit on things that are more likely to be important, right? So for example, 90% of my expression was made up by like 12,500. So we're getting a more realistic number that represents probably the transcripts that are mostly expressed by these sequences, these glands rather, sorry, right? And because they're highly expressed, they tend to be better assembled as well. So again, for those of you who do that thing, this is kind of an alternative approach to looking at transcriptome quality metrics. So we get a sequence that we want to figure out what it actually is, right? And we plug it into our database, and we're looking for things that are related to it based on some models of sequence evolution, right? And we can plug this into our database and say, hey, actually that right there is a transcript that encodes the full length of Cedefin precursor-like factor. Hey, we kind of would have expected to see one of those, right? What's that doing in the cloacal glands? Well, that's the ancestral gland for this, right? So it should have been there. And because I'm interested in proteins that end up making it to the outside world, right? Putative pheromones, candidate pheromones, we can also look at what's called a signal peptide, or actually really also other indications that the protein's bound for the outside world, right? So we can use this to say, hey, all right, I got tons and tons of data, thousands of genes, but let's start zoning in on things that might be interested. So we could look at how many of them were homologous and how many of them had a signal peptide, how many had both, right, out of our 90,000+ transcripts. And of course, when you do this kind of thing, most of them have neither, because you're doing this from scratch, right? And this is actually a really big number. 75% didn't have any homology or a signal peptide. But that's actually a number that crops up quite a few times in many of the de novo assemblies I've seen, actually. It's pretty much par for the course. And the good news, if you think about how big this number is, right, that means that for thousands and thousands of these things, 25%, we have a good idea of what it might be doing, right? So we have many of them that we found some homology for, right? Many of them that have a signal peptide, but we didn't find any homology. But that's a good indication that it might be a real protein sequence, right? It's actually sensical. It has a real signal peptide cue. But we also found a bunch that had both, right? I knew what they were and have a signal peptide. And again, this is actually an underestimate because you can use some of this information, even if it doesn't have a signal peptide, to say, hey, it's also probably bound for the outside world. So we end up with more good candidates than that even. And what can we take away from this, right? Well, we found a lot of known pheromones. Plethodon modulating factors, precursor-like factor. These were all over the place in these tissues, actually, which is neat because we've always thought of like PMF and PRF, I should have said as well, as being mental gland-specific pheromones, right? And actually in the eastern plethodon, people had hypothesized that SPF was kind of on the way out. It was being replaced by that new gene, PRF. But that's in the mental gland, right? The ancestral state was in another gland. So it could be that it's still super important for them. Lots of other cool things that we know about or are suspecting to be pheromones in these systems from other studies that I didn't really talk about and won't. Some others from different species as well. But we also found a lot of things that kind of, again, this is where we're starting sort of de novo, say, all right, what might be interesting that we don't really know about yet? What could be a putative or a candidate pheromone, right? And this is very hypothetical in this case. But let's start looking at what these glands were producing. Lots of carbohydrate binding proteins all over the place, actually. And this is really not surprising because some of the only biochemistry that had been done on these other glands was to basically just take sections of the tissues and stain them with some dyes that tell you kind of rough chemical classes that are present in there. And the PCG, the postcoital gland, and the cloacal glands are making lots of carbohydrates that could very well be serving as signals, right? And so there's lots of proteins that are probably involved in helping to produce them and helping to sort of get them out and maybe modulate that signaling in some other way. Lots of protease inhibitors. Again, we know from the mental gland these play a big, big role in the production of these pheromones. We've got to kind of make sure they don't get broken down. Maybe if you're secreting a territorial scent mark, right, you want to put some proteins in there that are going to help your other little protein buddies out, right? Keep them from being degraded. Toxins and venoms, which are important for amphibians as well, really toxins in this case, but we had a lot of homology to venoms as well. And that's important for salamanders, which are kind of noxious, but when you eat them, but which I haven't done. But also PMF, for example, would have annotated as a toxin, but it's been co-opted in this family of salamanders to be used as a pheromone. So maybe there's more going on there as well. Hormone-like proteins, reproductive-like proteins, immune-like proteins, lots of cool things that might be important. Plus lots of other things that might be interesting to other people, right, or myself in the future. And so we can look at the levels of expression. I'm going to kind of gloss over a lot of this stuff now for the sake of time, but let's just look at like the known plethodon pheromones and their level of expression, same plot multiple times for the different proteins. Its level of expression in each of the three glands. And this was a neat finding. PRF and PMF are actually being produced a lot in the cloacal glands at the transcript level. So there's a big caveat here, right? What matters for functionality is whether these are translated into proteins, which may or may not be the case. But we're seeing big transcript signals for them, especially PMF. SPF also showed up kind of all over the place. And these are actually underestimates because it's sort of a nuanced thing, but there's actually a lot of ones that were probably these things but didn't get assembled very well, other types of the sequence. So the real numbers would probably be much higher for these based on incomplete assemblies. But these are just like good, known, all right, that's an SPF sequence. And then some of the other ones that we didn't really talk about, I'm still going to click. And okay, so you can also do this kind of for all the different transcripts at once. And again, I'm going to move kind of quick here, but you can basically just say, all right, how much were these transcripts upregulated or downregulated in a given tissue, either the CG or the PCG or the DTB? And then you kind of cluster them based on similar patterns, right? Like high in the CG and then low in the other two. And you can build these into these kind of clusters and say, all right, well, let's look at this group that seems to have a sort of a high level of expression in the CG and lower in the other two, right? So you could cluster them into ones that are kind of related to each other and look at their expression profiles overall and figure out what's actually in these different groups, right? We're not going to go into it all because we would be here for the rest of eternity. But what can we take away from this, right? What did we learn? Well, I should say first and foremost that this was kind of meant as a first step, right, to enable proteomic observations, because again, that's what's going to matter for functionality. But we can also just take a look at what's in there, right? What transcripts are they making? And it's the first in-depth look at these things in a really attempted, anyways, at a comprehensive way to look at these glands and what they're making of these key signaling glands that people have been saying since the kind of mid-'80s, hey, we really need to figure out what these things are making, right? So we're doing that now. We're making some progress. And we found a lot of new proteins, and they might be involved. Of course, we need to experiment. They might be involved in these things that we know these glands are involved in, right? Reproductive signals, territorial, other types of social signals. And there's evidence for differential expression between glands at the transcript level, which you would also sort of predict. And mainly, it's a major step forward towards functional experimentation, right? We can start doing some of these proteomic work now, in addition to this work, to look at what proteins are being produced by these glands. And then, what might they be doing? So, to kind of bring it all home, what can we really just take away? Let's try to synthesize these different levels of scale and sort of put it all together. Well, we explored signal variation within the body of an individual, zoomed out to between members of the same species, and then between actually even populations of the same species, and between species, and between whole lineages of the entire plethodontid phylogeny, right? So we sort of span multiple levels of scale, and what did we learn, right? Well, within an individual, there's multiple signaling glands involved in different functions, right? And there's substantial variation between glands in terms of what the behavior is, and then there's some evidence now that also at the transcriptomic level, there's some variation there. And we could hypothesize here in italics that specific proteins in these glands are adapted for different functions, right? But we need to take this further. We need to use this transcriptome to help kind of enable the proteomic work. I should say you can do this de novo as well, but it helps to have sequences to which you can map these fragments of the peptide back to. So they work very well hand in hand. And we can then carry out behavioral and physiological experiments to figure out what are these things. All right, I think it might be a pheromone, but what's it doing? How does it affect receivers? Within a population, so a single population of the same species, we can see that there's substantial individual specific pheromone profiles, right? Due to things like variation in the isoform number and actually their sequence itself, what it is, and how it differs from other relatives of it. And there's variation in the levels of expression as well. And so therefore we could hypothesize the signal variation could have functional consequences. And so we need to take this further and test that hypothesis, right? We can characterize the effects of signal variability that we've been talking about here and figure out what do they do to receivers? How do they affect the way a receiver senses those based on how these different combinations of isoforms actually affect their responses, either behavioral or physiological, and how different levels of expression actually affect that as well. I'll tell you that our research group has done this, actually. We've looked at the way that different combinations of PMF in particular stimulates the vomeronasal organ, and actually what it does to courtship as well. And in order for appropriate functionality in terms of the behavioral response we would expect to see, you need these massive combinations of isoforms. When you start taking them away, you don't see the appropriate response of that reduction in courtship duration. We also see that different combinations of isoforms stimulate more neurons in the vomeronasal organ, a part of the olfactory pathway that these affect. So variations actually already seeming to be pretty important, as we would have hypothesized, right, as we did. I'll zoom out even further, right? Different populations, different species, different lineages. Well, we know they actually have evolved different pheromone components at different times, and there's differences in their levels of expression in these glands. Kind of mostly talking mental gland here now, right? And they have different isoforms of each component. And we can hypothesize, and actually this one's even really just mostly an observation, right? These lineages are diverging in their reproductive pheromone composition. And we need to, of course, do some more work to figure out, in this case, really, what are they even targeting? We still don't really know. We know they're in the olfactory clade, hitting the olfactory group, hitting these vomeronasal neurons in the olfactory chamber, but we don't know what receptors they're targeting. And we don't know, of course, what the effects of these sequence and structural variations would be on those receptors, right? So there's a lot of cool stuff to be done. But I want to just sort of zoom out even further here, right? I'm going to just drive you crazy with this. Luckily, we're at the end. But the thing that just fascinates me about life, and kind of why I like teaching so much, is you can really sort of take a bigger picture approach of things sometimes, is life is built of complex systems that are nested within one another, right? And if you're like me and you're interested in an organism or just behavior of organisms, what they are, the things that we can see, the creatures running around the world, right? You need to think about how that evolved, right? What are the effects of that behavior on the other individual entities that it's interacting with, right? And we also need to think, what are the causes of that, right? We need to say what lower level processes are involved in sort of generating that behavior. And this entire hierarchy, in let's say one case, is just one snippet in time as well. This entire multi-level complex is also moving forward in time. And they start to sort of diverge away from each other, and diverge away from each other, and diverge away from each other, to form what we know of as the tree of life that we are all a part of, right? And so in order to study evolution, which is really what we're doing here, you need to think about all these different levels and how they interact with one another. Let's just take one example, right? PRF glycosylation is a biochemical level trait on this protein. Specifically, it's a post-translational modification, sort of even above the level of solely the genes, right? But it's correlated with a transition in a behavioral trait, right? When we had that transition to olfactory delivery, there had to be changes at the corresponding level of the proteins, right? So there are multi-level sort of dynamics here in taking place in the study of evolution. And that is exactly what integrative biology is all about, right? That's what we're talking about here, taking sort of a bigger picture approach. And that's why I'm so proud to have been a part of this department. And that's basically what I've got for you. I should acknowledge a few people here quickly, mainly my committee, to whom I'm very appreciative for all of their time and great input over the years. Of course, the IB office staff has been awesome. I mean, I'm really not just saying that because everybody says that. Everybody says that because they're really awesome. And tons of other graduate students. I'm really only listing those that I've talked to you kind of at length about this specific research, but there's tons of others. You all know who you are, and all of you, thank you so much. Other faculty that have been really sort of instrumental in helping me become a better teacher and giving me other great opportunities. My collaborators. This work was a very collaborative endeavor. A lot of that biochemistry stuff, I never really claimed to be a biochemist, right? So I had a lot of help in making some of my ideas become a reality. Funding sources, right? And especially family and friends and everybody, many of which are here today. And the amazing salamanders of the world I've known and loved and admired, right? So with that, I'll take any questions. And again, thank you so much for coming.
All right,
Okay.
so I will ask the members of the committee to withhold their questions, but anybody else who might have questions? Have at it.
Yeah, Trevor.
So did you notice greater conservation at putative binding sites and glycosylated sites in PRF?
Okay. Do you have a second part?
And if you did see variation there, was that reflective of the species composition?
Yes, and both of that had been done before I even came along. So some of the work that really inspired a lot of this in PRF was by postdocs here, actually, and they looked at all the different sequences. Richard Watts, another actually PhD student, Kat Palmer, did some really cool stuff with this. And so they had looked at these sequences and actually tested these statistical hypotheses of evolutionary signatures, right? So I didn't do any of that directly, but they had done that stuff. Okay, I will. I thought you were plugged in, but so yeah, they had, and there's regions of the protein that definitely show signs of positive selection, actually very strong positive selection, but other regions of the protein, as you would expect also, are conserved, right, to maintain that same core fold, for example. And we kind of saw that in the blues and the reds, right? Some are pretty conserved, and some are really variable. And yes, their paper actually lined up all the different, that Watts paper in particular, lined up the sequences and with some of the ancestral ligands, the ancestral proteins, and showed that, yeah, these variable regions are right in binding sites. About half of all the variable regions they found were right in binding sites, which you would actually hypothesize that if you're talking about how these things are diverging away, right? There's a coevolution of males and females here because the receptor in this case is in a totally different organism, which is kind of neat. And so they're involved in this, and the hypothesis is that they're kind of in this this dance together, sort of evolving away from each other. And so the receptor binding sites are going to be important. Really, really good question. Thanks. Yeah, Phil.
Hey, in these species where you found subtle variation between the populations, do you or your colleagues have plans to look at the behavior? Do you expect to find little behavior differences through the population?
So actually one of the things that's neat about this and was sort of the hook for some of these early publications is that the behavior, with the exception of the two major lineages, the behavior is very conserved. The courtship ritual looks very much the same. Again, there's differences in the dermal delivery versus olfactory delivery, but the behavior is very conserved. And so actually that Watts paper, that was kind of the way they sold it, was, hey, there's a decoupling of these different levels here. Behavior is conserved, but there's rapid positive selection at the level of the proteins. So not really, actually. It seems to be doing similar things across the lineages, but at the molecular level, they're evolving very rapidly. That's an awesome question. Yeah, Emily.
Really nice talk. I was curious if there had been any studies looking at population genetic structure or the population.
No, is the short answer. Yeah, and that's like the most outside of my comfort zone too. And no, not really. But we know that, again, there's multiple alleles within a given population. That's definitely true. But we also know that, so actually let me finish that thought, so when you're looking at like the whole species, some of those are going to be allelic variants, but we also know when you sequence individuals, there's multiple genes present in there. They have many paralogs within there. So the population level thing, population genetic stuff is a whole unexplored realm. Yeah. Yeah, great question.
Okay, maybe one more question. We're running a little short on time.
Yes?
So have you seen anyone looked at seasonal variation in individuals in pheromone production?
Good question. So for one, the mental gland in particular actually only hypertrophies. It grows like into that big, in the olfactory group, into that big tumor-like thing. It does that every mating season. And so they're not producing mental gland pheromones in the off-season, if you will. And then they ramp up protein production and make as much of that stuff as they can, right, at a certain time of year. Beyond that, not so much. But one of the things that I got really interested in this in my master's was looking at actually things like condition-dependent signaling. I looked at the effects of dye quality in particular, and I wanted to know because they can communicate that. And actually I showed that they can detect changes in the energetic state within like 1 week of an individual. And females can actually sense this relatively rapidly, actually. And so I want to know, how do things like condition-dependent things, like I'm just navigating the world, more plastic traits, right? How are they communicated via chemicals? And it's a really interesting thing. So along with seasonal variation, there's like so many other ways that we could look at this, and that's a really good one as well.
All right, thanks.
Yeah.
Thanks very much. The committee, just follow the IB folks down to the quarterly 3030, and
Yep.
we'll reconvene there. Thank
Yep.
you, Adam. Great job.
Okay. Thank you again. All right. Thanks. Thank you.
Great. So I guess this